Hi there, my name is Mikey McGuire, and I'm going to be talking to you guys about Chapter 12 of our book, Art Harper's Illustrated Biochemistry, which covers biological oxidation. Biological oxidation is our main source of energy, and we're able to leverage redox reactions using the ETC and CA and the citric acid cycle to create 25 times more ATP than what we would create like anaerobes using just substrate level phosphorylation. Reducing equivalents have a diverse application in biology, and they deliver energy in the form of redox potentials. The common uses of redox reactions include energy production, biosynthesis, and detoxification. We can use it for beta oxidation, glucose oxidation, as well as NADPH, which feeds into the fatty acid synthetic pathways, as well as cholesterol synthesis. And we can use NADPH for reactive oxygen species reduction and liver-mediated drug metabolism. Redox reactions are always paired, so if we have a loss of electrons, we've got to have a gain somewhere else. The loss of electrons is called oxidation, the gain of electrons is called reduction. It's like handing off a baton in a relay race. There are two perspectives to look at redox reactions through. The first perspective just follows the transfer of electrons between substances and is a more fundamental chemical view, which is great for analyzing mechanisms of reactions. We can also use the biological perspective, and here we can track hydrogen as they're moved from one molecule to another. Hydrogen acts as a great proxy for electron transfer because it has low electronegativity, so whatever it's bound to, it's going to be basically donating its electrons to increase that bound compound's electron density. This is just a more practical approach for biological systems. We can quantify these redox reactions using redox potential. If you see a redox potential where E naught prime is greater than greater than 0, the compound has a tendency to gain electrons or be reduced. It's got a high affinity for electrons. If you see a compound with a redox potential E naught prime below 0, it has a tendency to want to donate electrons in a lower affinity for electrons. We can calculate the change in energy, the transfer of energy. It's called the difference in redox potentials, and it's acting like an anode and cathode of a battery. Basically, we use voltage to measure the redox potential, and a large positive difference shows a favorable transfer of energy, whereas of electrons, whereas a large negative shows some kind of energy barrier that needs to be overcome for the electron transfer. Redox potentials are split into two half reactions corresponding to each compound involved, being the oxidized compound and the reduced compound. Here we can see that oxidation of NADH gives us a redox potential of 0.32, whereas the reduction of molecular oxygen into water and the gain of electrons has a redox potential of 0.82. We can calculate the change the difference in these redox potentials using the delta E equation. The change in redox potential between two systems gives us an idea of whether a reaction is favorable or not. So here, we're going to go ahead and take the acceptor, which is our oxygen being converted into water, and then we're going to take our donor, which is our NADH, which is converted into NAD+. This reaction gives us a net redox potential of 1.14, which indicates that it is a favorable reaction in standard biological conditions. Gibbs free energy is analogous, but they are not identical. In fact, they have an inverse relationship. You can see that in the equation that we use to convert between net redox potentials and Gibbs free energy. You can tell the negative sign inverts the redox potential, so a positive redox potential gives us a negative delta G. The N is the number of electrons transferred in the reaction, and F is Faraday's constant. Just as delta G is standardized for biological conditions, we've got to do the same thing to redox reactions. So if you see an E naught prime, it's because we're looking at a reaction under standard biological conditions where the pH is 7 and the temperature is 25 degrees Celsius. Spontaneity can be inferred from delta E because of the relationship between the two. So a positive change in a positive difference in redox potential indicates a favorable reaction. And you can see that this graph right here is actually descending from top to bottom. So ascending from top to bottom. So we have higher negative numbers up here and higher positive numbers down here. We can actually see that the change in energy, or the change in redox potential between NADH and QH2 is around estimating 420 millivolts. We can also see that as this ascends, this is descending, creating a more negative delta G and a more favorable reaction. Let's actually evaluate this reaction just to see how accurate this graph is. So the half reactions involved are NADH to NAD and Q to QH2, ubiquinone to ubiquinol. The acceptor has a redox potential of 0.1, the Q to QH2, whereas the donor has a redox potential of negative 0.32. Together, these give us a difference in redox potential of 0.42, which is actually 420 millivolts. This justifies the graph, and also we can see that when we have a negative delta E, or a positive delta E, we're going to get a negative delta G corresponding. Using the equation to convert the two, we can also evaluate the reaction coefficient. The reaction coefficient is basically the relationship between substrates and products, and which side of the equation is preferred when it is gone to equilibrium. Here we can see the interconversion of the two, and I just really want to point out that concentration has a influence on whether reactions go to completion or not. We can classify oxidoreductase enzymes, which catalyze redox reactions in a couple different ways. They facilitate the transfer of electrons from one compound to another, and they also are capable of coupling favorable redox potentials with unfavorable redox potentials. And then they're also capable of coupling two unfavorable redox potentials, and we'll see that in this isocitrate dehydrogenase reaction. The half reactions of both of these substrates gives us two unfavorable redox potentials, but when we combine them into the equation, we see that there is a net positive redox potential, even though we're fighting uphill on both of these. So we can also see that redox potentials can use concentration to overcome an energy barrier. Here we're going to look at the malate dehydrogenase reaction. The half reactions both have negative or unfavorable redox potentials, and they create a net negative redox potential. However, our body leverages Le Chatelier's principle because our body has a ratio of 10:1 from malate to oxaloacetate in their cells. They also have more NAD plus than they do NADH, so these two concentrations actually drive the reaction to overcome this unfavorable energy barrier. There are a couple different types that I'd like to talk about. So first off, we've got oxidases, next we've got dehydrogenases, we've got hydroperoxidases, and oxygenases. First off, let's talk about oxidases. Their function is to pass the baton to oxygen. And you can see that right here. We're actually creating we're reducing molecular oxygen to H2O by donating electrons from A. Two common prosthetic groups are flavoproteins and heme-containing groups like cytochrome oxidase, which is complex 4 of the electron transport chain. Dehydrogenases pass the baton to a different compound than oxygen. So basically, if you see a reaction passing hydrogen or electrons to something other than oxygen, it's most likely a dehydrogenase. A couple of common coenzymes that they leverage for reducing equivalents are FAD, NAD, cytochromes, and ubiquinone. Together, these two types of oxidoreductase enzymes actually make up the electron transport chain. They use complexes 1 through 3 use reducing equivalents as electron donors. Complex 1 and 3 actually are capable of pumping protons using these redox reactions into the inner membrane space. And then complex 4 is also capable of pumping protons using the reduction of molecular oxygen into H2O, into water. Again, the difference is that a oxidase is passing electrons to molecular oxygen, whereas a dehydrogenase is passing them to a coenzyme. Hydroperoxidases use the electrons from peroxides or reactive oxygen species like glutathione to gain or lose electrons. These are important in reducing reactive oxygen species, which can create an oxidative cascade and will just go out and start either donating or stealing electrons from other compounds. There are two main types, peroxidases and catalases. Peroxidases oxidize hydrogen peroxide or other reactive oxygen species by passing electrons to some kind of oxidizing agent. A couple common reducing equivalents that accept these electrons are ascorbate, quinones, and cytochrome C, and a common prosthetic group is a protoheme. Peroxidases are super interesting in that that's the next slide. Red blood cells leverage these reactions to prevent the accumulation of reactive oxygen species. And one interesting disease is favism, in which the pentose phosphate pathway enzyme glucose 6-phosphate dehydrogenase is either inactive or it has a low activity, creating a reduced amount of NADPH in red blood cells. These red blood cells are less capable of fighting reactive oxygen species using this pathway here with glutathione peroxidase. And this will end up causing hemolysis, and in some Mediterranean versions of the disease, this can be fatal upon ingestion of fava beans, which have a high concentration of reactive oxygen species. Catalases are the incredibly efficient enzymes that I was just trying to refer to. So catalases are super important because they're able to actually use hydrogen peroxide as both the electron donor and the acceptor in these reactions. And so what they're doing is they're actually combining 2 molecules of hydrogen peroxide to make 2 molecules of water and 1 molecule of oxygen, which are less reactive oxygen species than hydrogen peroxide. They're found in many different tissues, and their reaction is one of the fastest known in the human body, allowing it to shred millions of molecules of hydrogen peroxide in seconds. Oxygenases are incredibly important for drug detoxification. We have 2 subcategories of these, monooxygenases and dioxygenases. Monooxygenases incorporate 1 oxygen atom from molecular oxygen into a substrate, whereas dioxygenases incorporate 2 atoms from molecular oxygen into a substrate. Here are a couple examples of monooxygenases. They're classified into a couple different classes, namely class 1 and class 2. And then there are also unclassified systems, which are very important in detoxification of drugs in the liver, which happens in the endoplasmic reticulum via the reduction or the oxidation of NADPH. Class 1 are typically found in the mitochondria, and they use a dehydrogenase which contains flavoproteins, whereas class 2 have a dehydrogenase called P450 reductase, which includes 2 different flavoprotein species to shuttle electrons over to P450. These are found in the smooth endoplasmic reticulum of many different tissues. Next, I'd like to talk about cytochrome P450, which is central in the detoxification of drugs. The cytochrome P450 is actually outside of class 1 and class 2 systems, the B5 system, and it improves the solubility of certain drugs, allowing us to excrete them in urine. and this allows it to dictate the duration of effects of many different drugs. There's also an interesting interaction in drugs like codeine, in which the hydroxylation by cytochrome B5 and P450 actually activate the drugs, and this is the case for codeine. The location of drug detoxification is in the endoplasmic reticulum of the liver, and B5 is just basically an assister to facilitate the transfer of electrons. Together, these compounds are responsible for 75% of our drug metabolism, and this is also why they are so important in dictating the duration of drugs. A couple drugs that they are capable of hydroxylating are morphine, oxycodone, and caffeine. And then they also can detoxify environmental toxins like carcinogens, benzoprine, which is a product of partially combusted organic matter. I found these resources through Indiana University, which gave me the types of drugs that are affected by P450, and then I found these different types of environmental toxins from the Department of Climate Change. Dioxygenases actually include both atoms of molecular oxygen into a substrate. One example is homogenatase dioxygenase, which is a key enzyme in amino acid catabolism. It's very important in the catabolism of aromatic molecules, including the amino acids phenylalanine and tyrosine, allowing them to enter our metabolic pathways. So I just want to peek at a couple dehydrogenases in metabolism. We've got glyceraldehyde 3-phosphate dehydrogenase, which gives us the reducing equivalent NADH. We also have some important enzymes in the citric acid cycle, which we already talked about, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Complex 4 cytochrome C is an incredibly important oxidase, which passes electrons from one copper center to two different heme groups, heme A and A3, which are referred to as cytochrome AA3. And then when molecular oxygen binds heme A3, the terminal copper center actually donates its electrons, producing a molecule of water. Oxidoreductases and P450 are incredibly important in steroid synthesis. We've already talked about how the product of steroid synthesis via CYP11A1 is going to give us pregnenolone, which is referred to as the mother of all steroid hormones. This P450 enzyme is responsible for the synthesis of every single steroid hormone in our body. And on top of that, the cholesterol used to create these steroid hormones are also produced from oxidation reduction pathways. There are many different fates of pregnenolone, namely creation of progesterone, cortisol, DHEA, or testosterone and estrogen. Here are my questions. Which of the following is an oxidase in the ETC? C4. Which of the following adds an oxygen to a molecule? We're going to see dioxygenases do that. Which of the following is important in the detoxification of drugs in the liver? We know that P450 is incredibly important for that. Which of the following has a spontaneous reaction in the forward direction? Only positive number here is NADPH to NAD+, so we know that must be the answer. Which of the following reducing agents are used for steroidogenesis and lipogenesis? We know that NADPH is used for these. Here are my sources. And I'm sorry I blazed through that. I know there was a lot of information, but I thought it was really important in the understanding of oxidation reduction processes in the human body.